Hello students, welcome to Raaonline. The chapter is Transport and Plant. The name of chapter is Transport and Plant under the section of Botany. It is question based brief discussion, just you can say crash preparation. Within short duration, how can you revise this chapter? Transports and plants. This is a plant. This is its subaerial part, that is root. Water and minerals. They are absorbed from the soil and they enter in the root and they reach to the vascular tissue, then transported to the other part of plant. Similarly, plant prepares food in the aerial part mainly by photosynthesis and green leaves. But organic matter is required by each and every part and each and every living cell. So this organic matter should be transported to the various part of plant. So this chapter basically deals with the transport of water, transport of minerals, transport of organic matter. And there are two types of transports which we see in the plant. Number one, short distance transport. This type of transport is cell to cell transport. In this case, vascular tissue is not involved. Short distance transport does not involve vascular tissue. Short distance transport does not involve vascular tissue. And the second type of transport, that is from one part of plant, for example root, to shoot or shoot to root. That is, this is long distance transport. And long distance transport involve vascular tissue, that is xylem and phloem. Long distance transport requires vascular tissue, that is xylem and phloem. And this is known as translocation. Long distance transport is called translocation, in which vascular tissue is involved. The question 1, it starts with that is the steroid hormones easily pass through plasma membrane through simple diffusion. This is actually with regards to the short distance transport. Plasma membrane is made up of lipid plus protein, mainly lipid plus protein. Lipid is bilayered. So lipid is bilayered actually. Lipid is bilayered, in which proteins are arranged in different ways. That is, some proteins are buried in lipid layer, some proteins are seen in the lipid bilayer, whereas others are seen over the surface. Lipid is bilayered actually, and it is in the form of quasi-fluid. So when hydrophobic substance or lipid soluble material can easily pass through lipid bilayer. Lipid soluble material can easily pass through by lipid bilayer by the simple diffusion process, that is according to concentration gradient. Number one, it is lipid soluble and the membrane is made up of lipid. So that's why they can easily be passed through the lipid bilayer. So first one, they can easily pass through plasma membrane. They can easily pass through plasma membrane because plasma membrane is made up of two layers of lipid in which proteins are embedded. Through lipid layer, lipid soluble material can easily pass through, but hydrophilic substance can't pass. In that case, specific protein channels are there. Protein channels are there for the passes of the lipid insoluble material or water soluble material. And in that case, you will see the two different types of transport. One is facilitated transport, other one is termed as active diffusion. So let's see if is there any question for that. So here steroidal hormones, they can easily pass through plasma membrane because plasma membrane is made up of bilayered lipid and lipid soluble material, but simple diffusion process can easily pass through. Then question number 2. Following criteria does not pertain to facilitated transport. Requirement of a special membrane protein, this is needed, this is required. High selectivity, yes it is. Transport saturation, yes it is, but it is not uphill transport, it is downhill transport. Facilitated transport, by the facilitated transport, hydrophilic substance enters through, hydrophilic substance can enter through or pass through membrane, but according to concentration gradient, according to concentration gradient, that's what is downhill, concentration gradient. Hydrophilic substance can pass through according to concentration gradient. So this is facilitated transport. It is not uphill, it is downhill. It is downhill through membrane protein, It is through membrane protein. If again concentration is there, then we can say it is uphill. And that in that case, energy is required and ATP serve as energy source. So active diffusion, active transport is uphill transport, whereas facilitated transport and simple diffusion both are downhill. Simple diffusion, there is no barrier, no proteins are required, whereas in facilitated transport as well as in active transport, specific proteins are required. But facilitated transport according to concentration gradient and active transport it is against concentration gradient. There is a difference between these two, that is uphill and downhill. This is the basic difference. And in uphill transport, energy is needed, it is not required in facilitated transport. This is the diagram. We can easily see your NCERT book and related with the facilitated transport. If only one molecule is passing, it is called uniport. If two molecules are passing through the membrane, one is in this direction, another one is in direction. First of all, if two molecules transport through membrane, this is called co-transport. Two molecules at a time can also be transported. This is called co-transport. If they are in opposite direction like this, this is antiport. This is antiport. For one molecule we can say uniport. For one molecule we can say uniport. But if they are in the same direction, then it is called symport. Now check the correct options. A is uniport, right. B is antiport, that is this one. And C is symport. So question number 3, its answer is C. That is, two molecules can also transported via through the membrane, or we can say the protein facilitated the transport for the two different molecules. If they are on same direction, then we can use the word symport. If they are in opposite direction, we can say antiport. If only one molecule transports, then uniport word is given. Question number 4. Cell increases its volume. This is cell. And its volume increases, that means definitely entry of a solvent in it. And this is possible when it is kept in hypotonic solution. Hypotonic solution, the solution whose concentration is less than the concentration of cell sap. What is hypotonic solution? The solution whose concentration is less than the concentration of cell sap. Such solution is known as hypotonic. That is, more solvent is more in this solution, whereas in cell sap solvent is less. So there is a osmotic entry of solvent in it according to concentration gradient of solvent only. That is, if a cell is kept in hypotonic solution, it takes water solvent from the solution and swell up. That is, cell performs end osmosis. If it is kept in hypertonic solution, cell performs exosmosis. That is, solvents are transported to the solution. In that case, if solvents are released from the cell becomes flaccid. But here the volume increases, that's why answer B is correct. In isotonic solution, there is a state of equilibrium. So neither in nor out. So correct answer for this question is hypotonic solution. Question number 5. DPD, that is diffusion pressure deficit. DPD. This is diffusion pressure deficit. Flaccid cell, that is no water in it. Turgid cell, that is with maximum water. Turgid cell, that is it has more water in that. So let's talk about the DPD, diffusion pressure deficit. If water is not there, then maximum diffusion pressure is there. If water is there, then minimum diffusion pressure deficit. Deficit denotes, that is lacking of what? Diffusion pressure. The solvent pressure diffusion pressure of solvent depends on the solvent only. Cell is flaccid, that is solvent is not there. That's why in flaccid cell the diffusion pressure deficit is maximum, whereas in turgid cell its diffusion pressure deficit is minimum. So minimum and maximum. Answer is, for this question, answer is maximum and minimum. First one. So answer is maximum in case of flaccid cell and minimum for turgid cell. If it is fully turgid, then 0. If fully turgid, then 0. So there is no possibility in this case. Question number 5 is over. Question 6. What will be 0 for fully turgid cell? Actually, fully turgid, there is a relation of dPd is equal to OP minus Tp. dPd is equal to OP minus Tp. And dPd is also called suction pressure. dPd is also called SPR, suction pressure. So see, if cell is fully turgid, that is with maximum water in it, in that case its OP becomes equal to Tp. In fully turgid cell, its osmotic pressure becomes equal to the turgor pressure, then cell does not require any more water. And if water is not needed, that is suction pressure will be 0. Fully turgid cell does not require water, that's why suction pressure will be 0. That is, water is already present in up to the maximum amount in it. So the answer is, for this question is, suction pressure or dPd is 0 for fully turgid cell. Question number 7. The process of reverse osmosis. Process of reverse osmosis can be discussed by this experiment only. That is, take a beaker, fix a semi-permeable membrane here, then here water is there, pure water is there, here solution is there, solution that is along with the solvent. Here solution is added, then you see pure water, nothing is present, that's why its osmotic pressure is 0. This osmotic pressure of this solution is 0. And whereas by the addition of solute, if solutes are there, there is some OP, that is some osmotic pressure. So that's why solvent enters in solution by the process of osmosis. Solvent naturally enter through in the solution by osmosis process. But this question is regarding reverse osmosis. That is, first of all, apply pressure over here to stop osmotic movement of solvent in it, and that pressure is equal to the OP. That pressure is equal to the OP of this solution. And if pressure is more, in that case direction of osmosis is reversed. If we apply pressure over the solution, apply the pressure over the solution, in that case the direction is reversed. In that case solvent moves across the membrane against concentration gradient, so that we can say this process is passive, that is, reverse osmosis is passive. That's wrong. It is active process. Pressure should be applied over solution. Question 8 now. Diffusion pressure deficit. Check the diffusion pressure deficit of cell A. Check the pressure. Then first one, that is cell A, dPd is 3. Cell B, dPd is 5. That is more dPd of a cell B. dPd is more in cell B. In cell A case, cell dPd is less. And the direction of movement of water is always from low dPd to high dPd. That's why the answer for this question is from cell A to cell B. Question number 9. Process of imbibition. Imbibition is property of hydrophilic colloid. Imbibition is property of hydrophilic colloid. If they are dipped in water, they swell. This is called imbibition. Imbibition is actually adsorption. It is adsorption indirectly. Indirectly we can say absorption, but it is actually adsorption. That is, molecule is stuck around this particular, water is stuck to the molecular surface only. And in diffusion case, if if a substance is added, initially it enters in the space, that is in between, space between two molecules. So it is a capillary action initially, then it is a simple diffusion process. So my answer is D for this question. That is, capillary action and diffusion both are involved. Which of the following is an example of imbibition? Uptake of water by root hair, exchange of gases in stomata. It is a diffusion only. It is a simple diffusion. Swelling of seed when put in the soil. Swelling of a seed when put in soil. Actually in seed micropyle is present. That is, a small opening is there. And through micropyle water enters. And why water enters? Because cotyledons are very good example of, they have a very good property of imbibition, because either they are made up of protein or they are made up of starch rich. In both the cases, that is, protein is also hydrophilic in nature, and similarly starch is also. So water enters in them and they swell up, and pressure exerted by the imbibition responsible for the rupturing the seed coat. So what is imbibition? That is, if the hydrophilic substance is dipped in water, it swells. That is known as imbibition. And swelling of seed is one of the good example. Question number 11. Pathway which is water moves through cell without crossing any membrane. That is, through non-living space. These are the two cells. This is cell 1 and this is cell 2, and this is the space between cell wall and the membrane. If water moves through this space without crossing membrane, it does not cross the membrane. Membrane is not at all involved. That is, movement of water takes place in between intercellular space as well as a space between cell wall and the membrane. In both the cases, water does not cross membrane. This type of movement is called apoplastic movement. This is called apoplast. In apoplastic movement, in apoplastic pathway, membrane is not at all involved. Water moves from non-living space only. So apoplastic movement, that is movement of water through non-living space, that is space between cell wall and a membrane, and second, the space between the two cell, that is intercellular space. This type of movement is known as apoplastic. When it crosses membrane, then it is symplastic. That is symplastic. So question number 11, answer is A. Passive absorption, the word as suggests. Passive absorption, that is, energy is not consumed. Passive absorption of water, that means root is not actively involved. Forces of absorption develops in the aerial part due to loss of water by the transpiratory activity. So we can say it is a result of tension on cell sap due to transpiration, increase respiratory activity, no. force created in the cells of root, no osmotic pressure in the shoot system, no. Better answer, tension on the cell sap due to the transpiration. In transpiratory activity, water is lost, so that forces of absorption in the area, develops in the aerial part, and the role of root is now passive. This type of absorption is termed as passive absorption. 13th question. Which of the following physical forces are supposed to be responsible for ascent of sap? See this one. Within xylem element, water forms continuous column. That is mutual force of attraction of water molecule. This is cohesive force. That is mutual force of attraction between water molecule. This is cohesive force. Second, the mutual force of attraction between water molecule and xylem wall. This is adhesive force. So there is a continuous column within the xylem element. There is a continuous column of water within xylem element, and two forces are responsible for that. One is cohesive force, other one is adhesive force. When there is a loss of water by transpiratory activity, so every cell maintains water and draw water from surrounding area. If water column continues, then water automatically lift up due to transpiration pull. So best theory which describes ascent of sap, that is transpiration pull and cohesion of water. If water column is not continue, then this transpiration pull will be useless. So main one is cohesion of water, that is mutual force of attraction of water molecule. That's why water column continues in the xylem element. 14th question. Lower surface of leaf has more stomata. This is seen in dorsiventral leaf. Dorsiventral leaf is either hypostomatic. Dorsiventral leaf is either hypostomatic or amphistomatic. There are two possibilities of dicot leaf. In hypostomatic leaf, stomata are present on lower surface only. In this case, only on lower surface. If amphistomatic, then both the surface has stomata. But in case of dorsiventral leaf, more on lower surface than upper one. In isobilateral leaf, almost equal number on both the surface. Number of stomata are almost equal in case of isobilateral leaf. Number of stomata almost equal on both the surface, whereas in dorsiventral leaf more on lower side than upper, because there are two possibilities. Hypostomatic, that is only on the lower surface, but if it is amphistomatic, then more on lower surface than upper one. Now come to the question number 15. Cohesion of water and transpiration pull theory operates only in passive absorption of water. Passive absorption of water. This is applicable for passive absorption of water or condition favoring transpiration. Conditions favoring transpiration. With respect to absorption, then passive, and with respect to the involvement of essence upset, then in this case that is conditions favoring transpiration. Transpiration pull theory operates only when conditions favoring transpiration. Then it is automatically lift up. That is question number 15. Column of water within xylem element. See this one. Column of water within xylem vessel in tall tree does not break. Reason: tensile strength of water. What is tensile strength? That is mutual force of attraction between water molecule. This is mutual force of attraction in water molecule, and number of water molecules are there. That's why its magnitude is very high. So that's why this column does not break. This is the tensile strength of water. Column does not break because of tensile strength. 16th question. Rupture and fractionation do not usually occur in the water column. Same question, but language is different. This is because of cohesion and adhesion. Cohesion, what is? That is mutual force of attraction of water molecule, and the force of attraction of water molecule and xylem wall or vessels, that is cohesion, and both are responsible for what? That is continuity of water column within the element. Then match the following, choose the correct option. Leaves, seed, root, aspirin and plasmolysed cell. First, leaves, they are concerned with the transpiration. That is, second one is right, because leaves have stomata. Leaves have stomata, and stomata are involved in the loss of water in the form of vapors. Then seed is directly connected with the imbibition, which we have studied in the past questions. Inside the seed, cotyledons are there. Cotyledons have either protein or starch, and that has the property of a imbibition. That is, they are hydrophilic. They take water, they absorb water by the process of imbibition. After second, that fourth option with the B. Then roots. Roots are directly concerned with the absorption. So with C, fifth one. Aspirin is anti-transparent. Aspirin, Dispirin and PMA. Aspirin, second Dispirin, third phenyl mercuric acetate. These are all anti-transparent. And what about the plasmolysed cell? Plasmolysed cell, they don't have water. That is, water is lost. That's why there is no osmotic potential. They have negative osmotic potential. So for E, that is third one. And in this respect, I think this is correctly matched. Option A. Option A is correctly matched. Question 18. Loss of water in the form of water droplets. This is seen in early, especially in winter season, early morning or late evening. When rate of transpiration is low and absorption of water is high. Rate of transpiration is low and absorption of water is high. Then minute pores are present on the margin. These pores are known as these marginal pores are known as hydathodes. These hydathodes have minute opening, and just below this opening space is there, followed by loosely arranged cells are there known as epitheme. These loosely arranged cells are known as epitheme. And this is the ends of vascular supply, that is xylem and phloem. This is ends of vascular supply. If rate of transpiration is low, if the rate of transpiration is low and absorption is high, root pressure increases. This root pressure acts on vascular tissue. So from xylem, water and minerals in this cavity, they enter in this cavity, and sap from the phloem. The water, minerals plus organic matter containing sap and minute droplets are released from the spores in early morning or late evening, especially in winter, by thin-leaved mesophyte plant. This process is known as guttation. It is seen in thin-leaved mesophyte. In which season? That is winter. At what time? That is early morning or late evening. And guttation is impact of root pressure. It's a manifestation of a root pressure. Also. And gutated water is impure. The released water is impure, whereas transpired water is pure. So this is the brief discussion of what guttation. The process of guttation takes place, that is by this one. See this one. When root pressure is high and transpiration is low. That's right. Absolutely right. Root pressure is low? No. Root pressure is equal? No. Root pressure is as well as transpiration both high? No. First is absolutely correct, that is absorption of water is higher than rate of transpiration. Then only root pressure increases, and impact of root pressure is seen over the guttation activity. Stomata open and close due to. Number one, depends on the guard cell turgidity. If guard cells are turgid, then stoma open. If guard cells are flaccid, stoma close. Because it is totally depends on the entry and exit of water. But no role with the root pressure, no role with positive pressure, no role with imbibitional pressure. Turgidity and flaccidity of a guard cell. Is mainly concerned with the stomatal movement, that is opening and closing. For opening they should be turgid, for closing they should be flaccid. Guard cells during day, let's check. During day, if transpiration is taking place, in that case there is influx of potassium, prime requirement. Potassium should be within guard cell. And second, uptake of chloride ions, this is also there. But main one is potassium, that is influx of the potassium in guard cell. Influx of potassium, because potassium, when it enters in the guard cell, it combines with the malate to form potassium malate. Potassium malate increases osmotic pressure. If osmotic pressure is high in guard cell, then end osmosis take place. If water enters in the guard cell, osmotically guard cells become turgid and turgidity favors opening. Turgidity favors opening. So during daytime, generally what is there? That is influx of potassium is in the guard cell. Valid explanation of movement of guard cell. Guard cells photosynthesis, starch hypothesis, best answer potassium influx and efflux. Influx favors opening, influx favors opening and efflux favors closing. Efflux favors closing. Efflux responsible for closing. When potassium enters in guard cell, it combines with the potassium malate to form potassium malate. It increases OP and OP draws water from surrounding cell and water enters in the guard cell by end osmosis process. And guard cells become turgid and turgidity favors opening.